Hello, good evening. Can anyone confirm? Am I audible? बताओ वॉइस सही से आ रहा है क्या? कोई कंफर्म करो बस. Okay, okay. We'll start in 1 or 2 minutes. शेयर कर दो सारे सभी फ्रेंड्स को. सबको बता दो अभी चालू. गुड इवनिंग, गुड इवनिंग. हम ऑलरेडी 2020 का पेपर डिस्कस किया था लास्ट वीक. ठीक है, आज से कंटिन्यूअस 5-6 पेपर है बाकी वाला, बाकी 2025 तक. ओके, वो भी खत्म कर देंगे. आई एम फाइन त्रिलोक. रत्नेश, हाय, हेलो, गुड इवनिंग. अभी सारे शेयर कर दो सबको. ठीक है, बस पारुल मैम का वीडियो देखते हैं आप लोग. हाँ, व्यूज बढ़ाते हैं उनका. हाँ, मुझे भी थोड़ा शेयर कर दो, मुझे भी बढ़ा दो थोड़ा व्यूज अच्छे से. इंफॉर्मेशन तो दे रहा है ना. गुड इवनिंग. मैराथन भी करेंगे इसके बाद. ठीक है, 5 ये वीडियो हो जाएगा, उसके बाद 3 मैराथन क्लास में खत्म कर देंगे प्लान बिल्डिंग. Sir, when will be the batch of 2027 start on the app? It will start soon after this particular exam. Okay, up to now, till now, you just finished the recording classes of this year. Okay, so that you will get in some basic idea. Live class will be starting from July or August. Okay, till now, you just complete the previous course content. Okay, you just digest that. शेयर कर दो फटाफट. अभी बस 35 दिखा रहा है. 50 होने के बाद शुरू करेंगे. इस पेपर में शायद 21. ठीक है, इन दिस पेपर मे बी अराउंड 20 क्वेश्चंस which are from plan bidding indirectly or directly. Some are from PGR, some are from IPR. Okay, and some questions will be coincidence with your CTEC, so we can't easily differentiate between the questions from CTEC and plan bidding sometime. Okay, maybe it's from floral biology, in those kinds of area, but around 25 will be the average questions from plant bidding. So as I told in our live classes also, and I am telling, okay, to crack those 20-25 questions means around 100 marks will be very easy. Okay, you are having some basic old and some concept about the subject plant bidding. Okay, you can easily score 100 marks from that part. Okay, that will definitely boost your rank. Okay, one who is having strong hold on plant bidding, they will be definitely be in the top rank. Okay, just follow my classes and we'll also do some marathon. Okay, one who are not in our course, okay, just follow these things now. Okay, and anyone interested to take or purchase our course, it is very, you are getting in very less price. Okay, you can purchase also. Okay, we'll start today's session. The way you clear the concept is amazing. Thank you, Trilok. सर इंग्लिश यस यस यस, सॉरी वो था हिंदी में आ गया था. मैं मेन इंग्लिश में भी बात करेंगे. ठीक है, as you guys already know about me, just I will introduce myself once again. I am Abhishek. Okay, PhD scholar from ICAR IARI New Delhi. I completed my undergraduation from VC Forum, which is under US Bangalore. I completed my masters from IARI New Delhi. Okay, I got All India rank 21st in ICAR PG exam, All India rank 4th in ICAR JRF or SRF exam. I also cleared ASRB net, joint CSIR UGC net, and I also visited Greece and Italy for one of my summer school in my PhD program. Okay, this is all about me. So first we will start with one beautiful quote from Dalai Lama. Okay, share your knowledge. It is a way to achieve immortality. Why I am telling just the one incident. Okay, when I am studying for this exam, ICAR PG 2020, that is one of my exam. In that, during that exam, one of my friend shared one video, simple video. Okay, someone is discussing some 20-25 questions. Okay, from genetics and plant breeding. That is from YouTube in that 2020. Okay, he shared me 3-4 days before the exam. In that particular video, from that particular video, okay, he is not preparing for ICAR PG that time. Okay, he shared that video. So I got directly 6 questions from that particular video. So the sharing is caring. So फटाफट शेयर कर दो वीडियो, शुरू करेंगे. आपको भी मतलब कहाँ से कुछ तो इन्फॉर्मेशन आएगा, ठीक है. If you are sharing the knowledge, so one or the other way you will also get some knowledge. That is my principle. Okay, just share the information what you know. Okay, this is the first question. Which of the following is form of executive conservation? They are asking about executive conservation. Okay, there will be 2 types of conservation, in situ and executive conservation. In situ conservation, okay, we will conserve the germplasms or genetic resources in their own habitat. Okay, the natural habitat. We are not disturbing it. We are not taking the germplasm from the particular place. Okay, we are conserving in their natural habitat so that we can allow the evolution in that particular place. But in case of executive conservation, what will happen? We will just move those germplasms out of their natural habitat, so we can store in the particular condition, maybe in low temperature, maybe in the good infrastructure. Okay, where there is an threat of extinction. Okay, due to the natural environmental condition. So we'll try to take those germplasms out of their natural habitat so that we can conserve them in the proper way. So अभी बताओ, which of the following is a form of executive conservation? इसमें executive conservation कौन सा है? लगा दो कमेंट में. सारे बता रहे हैं ये बी E. ठीक है? Yes, exactly. This is your first option. Your gene banks. In case of gene banks, you can see, okay, field gene bank, seed gene banks. These are the 2 important gene banks. Field gene banks, nothing but you can grow the crops or plants or trees, okay, in the field so that you can maintain them for generation after generation. But in case of seed gene bank, you will store them as a seeds. Okay, again there will be 3 kinds of storage in seed gene bank. Tissue culture repository. In that particular technique, what we will do? We will store them as a meristem culture, ovule culture, okay, all other organ culture, okay, in the cryopreservation or in the slow growth culture in the petri plate. Okay, if you have seen the banana tissue culture, okay, you can get a plant from that particular tissue culture so that you can conserve them as a tissue. Okay, this is your tissue culture repository. Again, it is ex situ. We are in the artificial condition, we are storing it. And your botanical gardens. Botanical gardens are nothing but one of the beautiful example. Okay, since I am from Karnataka, your Lal Bagh Botanical Garden. Okay, it contains so much varieties, a mixture of vegetation. Okay, so we are conserving them in the ex situ conservation. And again, there will be one botanical garden in GKVK Bangalore. Okay, these are kinds of examples of your ex situ. And national parks and biosphere reserves. What exactly the difference between national parks and biosphere reserves? We will see here. The national parks are, okay, the in situ conservation technology where we will restrict the people. Okay, we will restrict the any other outside interaction with the particular environment. Completely will restrict. Okay, in case of national parks. But in case of biosphere reserves, okay, there will be 3 boundaries, okay, inner core zone and intermediate and outer zone. In outer zone, we can allow the social, means the local community to use, okay, some for fodder purpose or for some the basic timber purpose, okay. We will allow some local community to use that particular resources. That is your biosphere reserve. But in case of national parks, you are completely restrict the outside people, okay. You are completely restrict the civilized means peoples, okay, in that particular national parks. You can see Jim Corbett National Park, okay, which is in Uttarakhand, which is very famous and first natural park, okay. Kaziranga National Park, which is in Assam. Gir National Park, Gujarat, which is famous for lions. Silent Valley National Park, which is in Kerala, and Bannerghatta, okay, Nagarwade, which is in Karnataka. Okay, these are the national parks, completely restricted, okay, from the outsiders. And your biosphere reserves, there will be some interaction between the local community and that particular area. Nilgiri Biosphere Reserves, which is in Tamil Nadu, Kerala and Karnataka, okay. Sundarbans Biosphere Reserves, West Bengal. You should know the difference between national parks and biosphere reserves, okay. Maybe they will ask some of the important national parks also or biosphere reserves also, so that basic idea you should know, okay. Nanda Devi Biosphere Reserves, which is in Uttarakhand, okay. Gulf of Mannar Biosphere Reserve, which is in which is in Tamil Nadu. Next, Botanical Garden, okay, as I already told, Lalbagh Botanical Garden, which is in Bangalore. Acharya Jagadish Chandra Bose Indian Botanical Garden, which is in Aura, West Bengal, which are some important, okay. National Botanical Research Institute, which is in Lucknow, okay. These are very important thing. You should know what are national parks, what are biosphere reserves, okay, what are botanical gardens, very important, ठीक है. इसमें एक तो क्वेश्चन आते रहते हैं इंस्टीट्यूट और एग्जीक्यूटिव में, ठीक है. इतना कॉन्सेप्ट होना चाहिए और एग्जैक्टली वो क्या है, ठीक है. कुछ कमेंट आ रहा है क्या? लालबाग इज द किंग. ठीक है. माय एंसेस्टर्स, रत्नेश क्या बोल रहे हैं एंसेस्टर्स बारे में? ठीक है, अभी आ जाओ. मैच द फॉलोइंग, ओके. वी विल सी द सेकंड क्वेश्चन इज योर मैच द फॉलोइंग, द वेरी सिंपल वन अगेन, okay. As I am again and again I am telling, plant breeding is very easy if you know the concepts, okay. If you know the facts, then it is very easy. You can see herbarium, the basic term, okay. If you see the herbarium, you can easily tell that collection of dried leaf specimen, that is or A4. Direct shoot, sorry it is B na, okay. Arboretum, what is arboretum? Botanical garden with trees and shrubs, okay. For A 3, there will be a 3 option. For B, as I told, 4. Then you can easily neglect these 2. Then these are the 2 remaining. You should always negotiate with the options, okay. There is no need to waste time, okay. If you find any one of the options which is correctly matching to any one of the options, then you can directly go for it, okay. हाँ बता रहा हूँ, सब बता रहा हूँ, ये ठीक है, आंसर तो यही है. देन बायोडाइवर्सिटी हॉटस्पॉट, ओके, द बायो जियोग्राफिकल रीजन विथ हायर बायोडाइवर्सिटी. अभी बताओ, कोई बताओ, ठीक है. आंसर तो ये, ठीक है. जर्मप्लाज्म्स आर नथिंग बट सम टोटल ऑफ जीन्स ऑफ ए स्पीसीज. आंसर तो ये मिल गया. अभी बताओ, कोई बताओ, इंडिया में कितना बायोडाइवर्सिटी हॉटस्पॉट है, वो कौन सा है, बताओ, कमेंट में बताओ जल्दी. इन इंडिया हाउ मेनी biodiversity hotspot are there? 4. अच्छा आंसर है. Yes, there will be 4 biodiversity hotspot, but we will consider 2 as a major biodiversity hotspot in the India, okay. Himalayan and Western Ghat. Yes, exactly. Himalayan, okay, and Western Ghats. These are the 2 important biodiversity hotspots, okay. You should know, okay, the very important Himalayan region of India and Western Ghats of India. Western Ghats of India will start from the Gujarat till the Tamil Nadu. Okay, Gujarat, Maharashtra, Goa. It will also cover Karnataka, Tamil Nadu, Kerala. Okay, this is your, in this particular region, okay, your Western Ghats will come from Gujarat to this part. This is your Himalayas. Okay, Indo-Burma, northeastern region except some Himalayan portion. Okay, again there will be Himalayas here, but this northeastern part we will consider it as a Indo-Burma. Okay, and Sundaland, it is the Nicobar Island. Okay, in Andaman Nicobar Island, again there will be a biodiversity hotspot. But whenever we are talk about the important biodiversity hotspot in India, in the prospect of world biodiversity hotspot, we will consider the major two, which is Himalayan and Western Ghats. Okay, these are the two important biodiversity hotspot in India. Okay, not Sri Lanka. We will not consider it in the case of India. Okay. Okay. The next question, again very simple. Okay, you just have to shoot one of the option. Don't, when you see this kind of question, okay, there is no need to confuse here, and there is no need to take stress also. अरे ये तो क्या दिया है, डबल टॉप क्रॉस दिया है, मॉडिफाइड सिंगल क्रॉस हाइब्रिड दिया है, अरे सारे छोड़ दो, आपको पता है टॉप क्रॉस हाइब्रिड. Okay, you know what exactly the top cross hybrid? It is just nothing but inbred into OPV. The simple thing, top cross is nothing but inbred into OPV. अभी ले लो, ऑप्शन ले लो, टॉप क्रॉस हाइब्रिड D inbred into OPV. Second option, you can just ignore this. Three-way cross hybrid, you know. Okay, in three-way cross hybrid, it is nothing but we will use just three inbreds. हाँ, ऑप्शन तो सी है, ठीक है. You can just see three-way cross hybrid. ये there will be three inbred A, inbred B into inbred C. Okay, for A3, in these three option, A3 you can only have, now you are having with these two option. Next, go for double top cross hybrid. Again there will be a word top cross. Again there should be a OPV for top cross. Okay, you can see there is a OPV, inbred A into inbred B into OPV. ले लो, C के लिए एक मिल गया आपका ऑप्शन. Means what it will do, it will help to choose your answer and also it will confirm your answer. Okay, if you go like this, it will do the both. Okay, that's it. In case of modified single cross hybrid, it is nothing but where there is a modification, where they are telling modification, you are crossing your inbred with the sister lines. Sister lines are nothing but again the same as your inbred A, but there will be some modification with respect to this particular A. So that's why we are calling it as a sister inbred. Okay, when you will cross this sister inbred with A, then with the inbred B, this is your modified single cross. Okay, what about double cross? Double cross is nothing but inbred A into inbred B. You will get single cross F1. Inbred C into inbred D, you will get again F1. Just you will cross these two, you will get F1. This is your double cross hybrid. In single cross hybrid, you will use two inbreds. In three-way cross hybrid, you will three inbreds. In four-way cross or double cross hybrid, you will use four inbreds. Okay, the simple thing. Top cross, if they are mentioning top cross, there should be a OPV. If they are using modified, then there will be sister lines. Clear? Batao? The same question was there in 2020 also. Yes. In plant breeding, okay, there will be a minimum 4 to 5 questions will be repeated after every year. Okay, heterozygous homogeneous, and regarding heterosis, regarding recurrent selection, these are very basic and important things. Okay, ले लो आप 100 marks. Okay, easily मिलेगा. ठीक है, समझ आ रहा है ना? ऐसे इजी क्वेश्चन आते हैं बस. अब कभी उठा के डालता है कोई क्वेश्चन, उसको किसी को नहीं पता है, ठीक है। उसको मतलब चाहिए तो हमारा कोर्स में हम सब कुछ कवर किया है, ठीक है। वो आउट बाउंड्री से आता है ना, आउट ऑफ द बाउंड्री समटाइम्स, ओके, वो भी हम कवर किया है ऑलरेडी, ठीक है। कितना लोगे? 53, बस 53 लगा दो, जल्दी शेयर कर दो। कोई मतलब किसी और नई ज्वाइन किया है तो बताओ, सबको बताओ, ठीक है। हम पारुल मैम वीडियो को बीट करना वाले हैं फ्यूचर में, बताओ, सबको बताओ। Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. In reciprocal recurrent selection, the two populations so developed can be used as synthetic variety. In the reciprocal recurrent selection. In reciprocal recurrent selection, the GCA of both population is improved simultaneously. बताओ आंसर बताओ। ये yes exactly the answer। अरे वीडियो ये लाइव में आपको मतलब मेरे साथ एंड तक रहना, ये एक स्ट्रेटजी दे देंगे मैं असर्शन और रीजनिंग क्वेश्चंस के लिए, ठीक है। आप किसी के प्रकार आप वो क्वेश्चन मतलब नेगेटिव मार्क आपका नहीं हो सकता है, ठीक है। एक स्ट्रेटजी बता देंगे, ओके। आई विल टेल यू वन स्ट्रेटजी, इफ यू be in the live session till the end, you will get one strategy regarding this assertion and reasoning types of question, okay. What exactly current, and also whenever I am discussing some questions, okay, I will not only cover that particular options or particular topic, okay. I will also give some glimpse which is related to, which is surrounded by that concept, okay. So that don't think that only we are discussing the simple thing, okay. One or the other way you are getting something else also, okay. What exactly the reciprocal recurrent selection, okay. This is your population A, okay. This is your population B, okay. What we will do? We will use two population simultaneously in case of reciprocal recurrent selection. Before that, you should know what exactly the recurrent selection. Recurrent selection is nothing but selection after selection. Selection, intercrossing, and then selection. This is a cycle, okay. In case of population improvement program, in case of cross-multiplied crop plants, we are always concentrated towards population, okay, not about individuals. In case of self-planted crop plants, what we will do at the end of the program, okay, whatever the thing, okay, we will start from the single line, then we will multiply the seeds of that particular single line. In case of your pure line, in case of pedigree, in case of backcross, okay, we will just try to identify any single line which is very best, then we will just multiply that seeds of that particular line. क्रॉस planted में ऐसे नहीं चलेगा, ठीक है। It's if you are again if you are trying to isolate the line or if you are trying to multiply the seeds from single line, क्या होता है बताओ, कोई बताओ अभी कमेंट में बताओ। What's the demerit if you do like this? If you are concentrating about only single line, as case of in case of self-planted crop plants, क्या हो जाएगा? What will happen in case of cross-planted crop plants? बताओ, I am expecting in only the single word. बताओ, कोई बताओ। बताओ बताओ जल्दी कमेंट में बताओ। आईडी ओब्बा सिंपल शॉर्ट फॉर्म exactly inbreeding depression, okay. That's why in case of cross planted crop plants we are targeting about the population, okay. We are much more attention towards the whole population, okay. We are trying to accumulate the favorable alleles in the population. Here in case of cross planted crop plants we are not into the single inbreed or single line, okay. समझ गए? इतना बेसिक कॉन्सेप्ट तो इतने है ये, मतलब इसी से अराउंड सारे कॉन्सेप्ट, ओके, इवॉल्व हुआ है, ठीक है। दैट्स व्हाई यू कैन सी द सिंथेटिक्स अगेन पॉपुलेशन, composite population, ठीक है। your recurrent selection, at the end of the day you will end up with the synthetics and composites, Hybrids are the different way, okay? You will get hybrids in both self-planted and cross-planted croplands. But apart from hybrid, it's all about the improvement of a population in case of CP. Okay, and also you should kept one sentence in your mind, that is accumulation of favorable alleles in the population. That is your main objective in case of cross-planted croplands. Favorable alleles का frequency increase. Yes. ठीक है. Simple recurrent selection में क्या करते हैं हम लोग? What we will do in simple recurrent selection? There is only one population. You will just select the superior individuals from the population. Okay. You will self those. Okay. You will self those particular plants. You will take that seeds, selfed seeds. In the next generation, you will grow those the superior selfed plants, and you will make the intercross among themselves. Okay, this is the basic idea in case of simple recurrent selection. You are not doing any crossing here. You are not testing with the OPV or with the inbreed. There is no testing for GCA and SCA in case of simple recurrent selection. What we will do? We will select some superior plants in the population. Okay, there is a maize plot. We will select some 20 maize plants which are very superior in case of their phenotype and yield. We will self that particular 20 plants. We will get cobs, 20 cobs. Then we will take those 20 cobs and we will sow in the next season. Then we will do the maximum, okay, the possible intercross between those 20 the 20 cob seeds, the plants. Okay, this is the first cycle. Then what we will do? Then we will again grow. We will select some, then the best 20 plants again. This is the cycle, which is in simple recurrent selection. In case of recurrent selection for GCA, what we'll do? Before selfing the seeds, we will also do the crossing with the open pollinated variety. Okay. This is your population. Okay. We will cross with OPV. OPV is nothing but open pollinated variety. It will give the different combination of gametes. Okay, this is your population. What we will do here? You will select some superior plants. I will select some 4 superior plants. I will self them. Okay, I will self these 4 particular crosses, 4 particular individuals, superior individuals, and I will also cross these 4 individuals with OPV. There will be a selfed seeds, this from 4 particular individuals, and also there will be a crossing with OPV. I will kept these particular selfed seeds, okay, separately. I will store. I will evaluate these particular crosses, 4 crosses. Okay, in the next season. In this out of 4 crosses, which is giving the best output, which is giving the best yield out of these 4 crosses. Okay, I will select only those selfed seeds. Okay, I have 4 selfed plants. Okay, I have stored it. Okay, then by seeing this OPV with a cross with OPV, okay, I will get 2 cross which are superior. Okay, they are giving superior progenies. Okay, for example, this one and this one. From these 4 selfed plants, okay, I will only select these 2 best plants. Okay, and I will grow in the next generation. Okay, that is this is in the first year. This will be in the second year. Okay, the evolution of this particular class. Then in third year, I will grow these only 2 individuals and I will make the crossing, intercross among these particular 2 individuals. This is the first cycle. What I am doing? I am accumulating the favorable alleles and I am developing the population. Okay, which are best GCA, general combining ability. Okay, when you are testing with OPV will it will give different number of gametes, the different types of gametes. Okay, open platelet variety. Okay, it is not an homozygous. It will give different types of gametes. So there will be different types of crosses. So when you will evaluate those crosses, okay, if the inbreeds or if the any lines which are performing well with the OPV, that means they are having good general combining ability. ठीक है, समझ रहे हैं ना? I will take those individuals which are having good general combining ability and I will make the intercross among only themselves. Okay, I will not take all the 4. This is the GCA. In case of SCA, recurrent selection for SCA, in the place of OPV, there will be a inbreed or single cross hybrid. This is homozygous. This is narrow genetic base. Okay, this is broad genetic base, your OPV. The same procedure again. Okay, you will cross with the inbreed or hybrid. You will evaluate those progenies and you will also self these particular seeds. Okay, which are giving the best performance with the inbreed. Okay, we will only select those selfed seeds. You will only sow those selfed seeds in the next season and you will make the intercross among them as well. First cycle. You will repeat it. Okay, up to the breeding objective, up to your basic criteria. Okay, you will do the cycles. This is your recurrent selection for GCA. But in case of reciprocal recurrent selection, what we will do? There will be 2 population. Okay, there is no OPV here. There will be 2 population. You will use this population as a tester for this particular population, second population, and this particular population as a tester for the first population. Okay, so that you can simultaneously improve the both of the population. Okay, यही है basic concept. आउटकम क्या है आपका रिकरेंट सिलेक्शन फॉर जीसीए में? आउटकम क्या है? इसके बाद आप रिपीटेड साइकिल बताओ, कोई बताओ, कमेंट में बताओ, व्हाट इज द आउटकम ऑफ रिकरेंट सिलेक्शन फॉर जीसीए? लगा दो कमेंट में लगा दो भाई. What is the outcome of recurrent selection for GCA? Maybe there will be a question they can ask. Exactly. Synthetics. अरे भाई ये नथिंग बट सिंथेटिक्स है. हम जीसीए के लिए टेस्ट किया है, ठीक है, इंटरक्रॉस कर रहा है among themselves. At the end of the day, we are getting the populations which are superior GCA. वही है ना आपका सिंथेटिक्स. इसमें बताओ रिकरेंट सिलेक्शन फॉर एससीए में बताओ, रिकरेंट सिलेक्शन फॉर एससीए में आउटकम क्या होता है बताओ. अरे भाई शेयर कर दो वीडियो, सबको शेयर कर दो. व्यूज ही नहीं आ रहा है, बस 53 है. ऐसे नेगलेक्ट करो प्लांट ब्रीडिंग के लिए, प्लांट ब्रीडिंग को, ठीक है. 20 क्वेश्चन 25 क्वेश्चन, हाँ पक्का 100 मार्क्स मिलेगा. अभी कॉन्सेप्ट तो मिल गया ना रिकरेंट सिलेक्शन के बारे में. हाइब्रिड वेराइटीज नहीं, हाइब्रिड वेराइटीज बाद में करता है. द एंड प्रोडक्ट ऑफ रिकरेंट सिलेक्शन फॉर एससीए इज inbreeds, the superior inbreeds which we can use for hybrid development. Okay, when you are crossing with inbreed, okay, you are lines into inbreed, you are testing for SCA. Okay, the lines which are giving the best specific combining ability, okay, you can select that from the population. You can use them as a inbreed for the hybrid development. रिकरेंट सिलेक्शन में क्या होता है? रिकरेंट सिलेक्शन, रेसिप्रोकल रिकरेंट सिलेक्शन में you are simultaneously improving 2 population. At the end of the day, okay, you are improving for both GCA and SCA. Okay, since for both the populations, okay, counterpart one or the other way, so that at the end of the day, you are getting a population which is improving for both GCA and SCA. इतना है कॉन्सेप्ट रिकरेंट सिलेक्शन में, ठीक है, कुछ नहीं. साइंटिस्ट तो देखो, ओके, आप बुक्स में भी देख सकते हैं कौन दिया है वो, ठीक है, वो उतना मतलब मेमोराइज्ड है, बस कॉन्सेप्ट तो इतना होना चाहिए। इधर देखो सिंपल रिकरेंट सिलेक्शन, पॉपुलेशन यूज्ड वन पॉपुलेशन, there is no tester needed. As I told, in simple recurrent selection we will only select the superior plants and we will just self them, okay, and after in next generation we will intermate among themselves, okay. The main improve, it will mainly improve the phenotypic performance. अभी देखो इधर recurrent selection for SCA, one population, the tester is broad genetic base, that is your OPV, ठीक है, OPV, okay, which is improving for GCA. Recurrent selection for SCA, one population again, narrow genetic base or inbred tester, it will improve SCA, okay. Here there will be a synthetics end product, here there is a inbred for hybrid development, okay. Here it will improve both GCA and SCA. Two population and reciprocal recurrent selection, each population test the other, clear. Sir, please explain how the recurrent selection for SCA gives inbreds. अरे बाबू देखो, what will do in case of recurrent selection for SCA, okay, you will cross the individuals, okay, the lines with the any inbred or hybrid, okay. Your inbred or hybrid are homogeneous population. Homogeneous populations are nothing but they will give only one kind of gametes, okay. Out of some 4, here there is a 4, but we will test the more number of inbreds or lines, for example 100, you will cross these 100 lines with the same inbred, ठीक है. सेम इनब्रिड से मतलब इनब्रिड तो क्या है, होमोजीनियस है, सब में आप एक ही गैमेट आ रहा है, ठीक है. देयर विल बी 100 possibility, 100 crosses, okay. From one plant you will cross with one inbred, you will get one, like that you are having 100 individuals, you will cross with the same inbred, you will get 100 crosses. Then you will start to evaluate those 100 crosses, okay. Out of 100, obviously you will get some 10 good inbreds which are giving superior performance. That means those 10s are having the best SCA, means specific combining ability with this particular inbred, okay. If this inbred is your parent B in case of hybrid development, the same inbred you are using, okay, as a another parent in your hybrid program, okay, and your 10 inbreds from this particular population are giving the best SCA with this. So out of this 10 you can release any 1 or 2 varieties as a hybrid varieties, ठीक है, समझ रहे हैं ना, बताओ. ग्राइफ इंदौर 1931, समझ गए ना, how we can use recurrent selection for SCA for inbred development or hybrid development. बताओ कमेंट में बताओ, समझ गए ना. In recurrent selection GCA what will happen, we will cross with OPV, but OPV will not give the same gametes, there will be a mixture of gametes, ठीक है, and there will be a different number of progenies, different number of crosses, so that you can only say that this particular inbred, okay, this particular inbred having the different, again there will be number of crosses, okay, since there will be number of different gametes from OPV, for particular line there will be a so many number of crosses, so that if you take the average of this particular cross you can tell the GCA, general combining ability of that particular inbred. But here if you cross with inbred you can easily tell that the specific combining ability of the individuals, okay, clear है ना. Yes, okay, this is all about your recurrent selection. You can see there is a simple question regarding recurrent selection, so I tried to cover it each and everything which is related to recurrent selection. This is the benefit what you will get. Sir, which is more practical, synthetics or hybrids? अरे भाई वो तो it will again depends on different crops and different conditions, okay, where you can मान लो आप farmer, ठीक है, आपको मतलब every year you are not able to buy a seeds, so you can prefer synthetics, and synthetics are very poor in yield compared to hybrid, okay, compared to hybrid which are very poor. but they will give good yield. Okay, but when you are want more yield and you are able to purchase the seeds every year, then you can go for hybrid. Okay, but practically we'll always go with the hybrid. अभी तो फार्मर के पास तो पैसा है भाई, सारे हाइब्रिड सी ग्रो करते हैं. ठीक है, कौन पूछा था क्वेश्चन? स्वर्णा. ओके, आ जाओ, इधर आ जाओ, थोड़ा जल्दी जाएंगे अभी. शेयर कर दो, वीडियो शेयर कर दो, सबको बताओ. Sharing is caring, sharing is caring. You should share the knowledge. Single cross hybrids can be used for a variety of purposes such as, बताओ, कमेंट में बताओ. सर प्लीज टेल मी प्रॉपरली इन हिंदी. हाँ हाँ पारुल. अरे हिंदी तो समझ आया ना बच्चा, आप बताओ. प्लीज यूज इंग्लिश सर. अभी एक कोई बताओ, मतलब मैं दोनों में मिक्स कर रहा है. ठीक है, मतलब समझ आ रहा है ना. मैं मेरा मोटा मतलब फाइनल आउटकम एट द एंड ऑफ द डे यू शुड गेट द कॉन्सेप्ट, वेदर इट मे बी इंग्लिश और हिंदी. गेटिंग राइट? कॉन्सेप्ट तो समझ आ रहा है ना? बताओ बताओ, जल्दी बताओ. ऑप्शन डाल रहे हैं फिर से. बाइलैंग्वेज इज ओके सर, मतलब हिंदी तो समझ आ रहा है ना, वो बताओ. पारुल को नहीं आ रहा है. यस सर, ओके ठीक है बच्चों, शेयर कर दो, शेयर कर दो. ओके, सबसे अच्छा व्यू आना चाहिए. ठीक है, single cross hybrid can be used for a variety of a purpose such as, okay, if you see the option, okay, again the single direct shoot, okay, back cross generation. अरे क्या भाई ये back cross generation. ठीक है, सिंगल क्रॉस हाइब्रिड में बैक क्रॉस क्यों आएगा? ओके, वही बैक क्रॉस विल कम इन सिंगल क्रॉस हाइब्रिड. यू कैन जस्ट इजीली नेगोशिएट इट. composite variety, अरे भाई composite variety will just intercross among the inbred lines. At that particular concept we are not going for the testing for GCA also. Okay, there is no way for the single cross hybrid in case of that particular development of composite variety. B और E निकाल दो, कहा B है, निकाल दो निकाल दो निकाल दो. A C D. ठीक है, आंसर तो C. हाँ C. I want both. Sir, your Hindi is so cool. Thank you यार. Can R3 you. पारुल देखो देखो. मेरा क्लास पारुल भी अटेंड किया है, बस इतना चाहिए मुझे, मतलब इतना चाहिए था बस. ठीक है, देखो इधर डेवलपमेंट ऑफ 3-वे क्रॉस हाइब्रिड मैं ऑलरेडी बताया था. A into B into C. You should need single cross in this particular thing. Development of double cross hybrid, A into B into C into D. You should again need single crosses here. Prediction of double cross hybrid very important. कौन दिया था prediction of double cross hybrid? बताओ. Prediction of double cross hybrid important. क्वेश्चन आया था एक बार. ओके, prediction of double cross hybrid. Jenkins, good. Jenkins given the prediction of double cross hybrid. There will be 3 different ways to predict the double cross hybrid. Okay, the performance of double cross hybrid, but the important one is the average of non-parental single crosses. Okay, again you need single cross for the prediction of double cross. Okay, अभी बताता हूँ मैं, simple. Okay, ये आपका, this is your double cross. Okay, you want to predict the performance of this particular cross. Okay, what they will give? They will give you the performance of non-parental single cross. Non-parental single cross are nothing but your A into C, A into C, A into D, A into D. This is non-parental again. B into C, B into C, B into D, they will give B into D. Okay, they will give like this. Okay, 6 kg, 6 kg again, 4 kg, 4 kg. बताओ, कोई बताओ, प्रिडिक्ट करो अभी इसका प्रिडिक्ट करो, डबल प्रिडिक्शन ऑफ दिस पर्टिकुलर डबल क्लास का परफॉर्मेंस प्रिडिक्ट करो, ये देखे सारे, बताओ बताओ, रिमी बताओ, आंसर बताओ। लगा दो यार, आंसर लगा दो। 5 5 5 5, ठीक है। इवर डबल क्लास हाइब्रिड का परफॉर्मेंस, ओके, नथिंग बट योर average of this particular non-parental single crosses, okay। B से टोटल एवरेज लगाए तो इफ यू टेक द एवरेज 5 kg, ये तो सिंपल है जेनकिंस का, ओके। एंड देयर आर अदर strategies also how we can predict. You can take the particular inbred mean, then you can take the average, okay। There will be different kinds of predictions, but the important one, okay, which is practical, is this thing, okay, 5 kg. Chronological steps in the development of hybrid, अरे ये तो easy है। आंसर आंसर, इसको आंसर बताओ, लगा दो यार, जल्दी लगा दो। बीच में एक इंपॉर्टेंट स्लाइड है, ठीक है, सबके लिए। देख रहे सारे, मतलब कौन सा चूज करना है, बस ऐसे ऑप्शन रहते हैं ना, देखो ये 3 में है, पक्का ये आप डायरेक्ट नेग्लेक्ट कर सकते हैं, ठीक है। 3 ऑप्शन कंफ्यूज करने के लिए दिया है, ठीक है। फर्स्ट यू शुड डेवलप द इनब्रीड, दैट इज योर फर्स्ट केस, ओके, योर ये। देन आफ्टर डेवलपमेंट ऑफ हाइब्रिड, व्हाट यू विल डू, you will do the morphological evolution or evolution testing for combining ability. What you will do, बताओ। First you should see the morphological evolution of that particular inbreed. What is morphological evolution, okay? If you are using as a female parent, that inbreed you are using a female parent, it should have good female characteristics, okay, that are very short in length, okay, so that your cob will get good pollens, means the effective pollens, and the cob length should be very maximum, okay. There should be a more number of kernel rows. These are some female characters. If you are choosing as a male parent, okay, what you have to do, you have to see for the plant height. If there is a good plant height, so the pollen distribution will be very good, okay. That is the main objective in, if you are using as a male parent, you should see for the morphological evolution of an inbreed, that is your D. Then after that you will check for combining ability, okay. When you are using as an inbreed for the hybrid development, you should test for the specific combining ability. First you will see for that ADC, then after development of hybrid you should check with the checks, okay, standard checks. There will be already some variety which are ruling the particular hybrid, which are ruling the particular year, in that particular year, particular area. You should always compare with that particular check, that is your B. Then seed multiplication, you go for seed multiplication and you can give the seed. ADC B, ADC क्या हुआ, ADC B, कोई बता रहा था B, कोई बता रहा था C, C will be the answer, okay. Again this will be our courses, okay. You are already getting our courses with 199, okay, 1999 for this particular exam, this year exam, Okay, the one who are not purchasing, you can just purchase it now and you can cover the entire syllabus within this particular 30 or 40 days. Okay, one who are preparing for test series, you can purchase our course, okay, which is 799 course for next year, but you are getting it for 499 for this particular year again. Okay, you can also purchase our test series. Then for next year ICAR PG exam for 2027, the course will be available for 4999. Okay, the one who are already purchased this particular our courses, they already know about it. You can just take the suggestions from those fellows and you can purchase. And for 2028 exam, the foundation batch, you are getting at 8999. Okay, you can also purchase that course. Okay, this is all about our courses. Okay. Next question. Heterosis cannot be fixed if it is obtained through. Cannot be fixed. You should always see the questions. Cannot be. Okay. Shoot लगा दो. Direct लगा दो. Development of F1 hybrid, development of inbred, development of synthetic variety. ये दोनों में नहीं हो पाएगा. Okay. You are not able to fix the heterosis in simple F1 hybrid. If you are developing your synthetic variety also, you can't fix the heterosis. Where you can fix? You can fix in apomixis, you can fix in balanced lethal system in case of vegetative propagation. A, B, E. लगा दो A, B, E. Sorry, you cannot be fixed. Okay. C, D, C, D, 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 D. अरे अभी इधर बताता हूँ. Apomixis तो पता है. ठीक है. There will be a complete omission of the sexual reproduction. So you can directly fix the heterosis. Since there will be no production of gametes, you can directly fix. But in case of balanced lethal system, what will happen? In balanced lethal system, okay, if you consider there will be two genes, AA, BB. Okay, when there is a condition of AA, BB, that is, if you take the homologous chromosomes, okay. अरे एक मिनट. There will be a B, B. Okay, this is the condition. For example, when this condition, at this particular condition, okay, your progenies will die since they are in homozygous condition. If it is condition is small a, small a, small b, then also your individuals will die. Okay, since again there will be a homozygosity. Okay, lethality. When there is an homozygous condition, they are going to die. But when the condition is capital A, okay, small a, capital B, small b, in this particular condition, okay, your individuals will survive. What that means? You can see the heterozygosity. Okay, they are fixing the heterosis. Always there will be three kinds of gametes. Okay, there will be three kinds of progenies. Capital A, capital A, small a, small a, small a. Out of this, okay, 25%, 25%, 50%. Always this will survive. These two will be leading to death. Okay, so from this method also, through balanced lethal system, example, which is the primary example, Inothera lamarckiana. There will be two types of gametes, Gordons and Levens. In that particular case, there will be two kinds of gametic proportion. When there is a hybrid between them, then only they will survive. Okay, when there is a Gordons and Gordons chromosome set, they will not survive. Okay, valence and in that case also they will not survive. When there is a hybrid between those two sets, then they will survive. Means in heterozygosity only they will survive. That is your balanced lethal system. It will also fix your heterosis. Vegetative propagation. If you are having an hybrid potato, okay, if you through sexual reproduction you will produce a hybrid. Then after that you will propagate through the vegetative propagation through the tubers. or in onion through on bulbs, okay, in sugarcane through the shoots, you can continuously maintain the heterosis. Okay, then there is no production of gametes, right? So you can maintain, and there will be always one confusion. Okay, what about polyploidy, sir? Yes, कोई लगाया है ना? Polyploidy also fixes heterosis, but in not always. Okay, it will not fix the heterosis always. Only in case of allopolyploids. Which is the example for allopolyploid? बताओ कोई? allopolyploid example बताओ भाई. अच्छा वाला wheat. Good, very good. You can see A, B, D, D. Okay. Only in case of allopolyploidy, your polyploidy will fix the heterosis. You can see there is a hybrid between A, B, and D. There will be 3 different genome. Okay. So we can consider it as a heterosis between A, B, D genome. Okay. You can also tell allopolyploidy fixes the heterosis. Okay, not in always in the same case. Triticale, triticale में, cotton में, ठीक है, when there will be 2 genomes, you can say that it will fix the heterosis. ठीक है? अरे भाई वोट्स क्यों बीच में आ रहा है? वोट्स तो डिप्लॉयड है ना भाई? समझ गए ना? वही पॉलीप्लॉइडी विल फिक्स द हेटेरोसिस. समझ गए ना? बताओ कोई बताओ. क्रिटिकल इज एम्पी डिप्लॉयड सर. अरे वो सही है, एम्पी डिप्लॉयड है. एम्पी डिप्लॉयड is nothing but first it is allopolyploid. Okay, ampy diploid is, it will come next. Okay, वोट्स बताया था ना. वोट्स मतलब बेसिक तो डिप्लॉयड है वोट्स, ठीक है, मतलब टेट्राप्लॉयड और इसमें बताया हुआ, इसमें भी आता है, वो तो बेसिकली डिप्लॉयड है, ठीक है. वेयर देयर इज एन allopolyploid, in that cases your ploidy level fix the heterosis, that you should know basically. ओके, ठीक है, जाता है नेक्स्ट क्वेश्चन. फॉर एस्टिमेशन ऑफ मिड पेरेंट हेटेरोसिस. मिड पेरेंट हेटेरोसिस में कल का क्लास में भी बताया था ऑलरेडी. ओके, we have already discussed in the yesterday's class. The genotypes include in the trials are, बताओ मिड पेरेंट हेटेरोसिस क्या होता है फार्मूला? F1 minus mid parent divided by mid parent into 100. Your heterobeltosis, बताओ heterobeltosis. F1 minus better parent into 100. Your standard heterosis, F1 minus standard check or national check divided by standard check or national check into 100. अभी बताओ, for estimation of mid parent heterosis the genotypes includes in the trials are, लगा दो क्वेश्चंस, लगा आंसर लगा दो, ऑप्शंस ABD only, ABC only, ABE only, BCD only. B A B C. Okay, for mid parent, okay, you should take the average of A and B. Average of A and B will give you the mid parent value. Then you should have the F1 hybrid, the value of F1 hybrid, so you can easily calculate the mid parent heterosis. Simple, ठीक है, आपको बस फार्मूला पता होना चाहिए. An artificial seed consists of a gel enclosing one of the following components. Again easy question, which is from CTech anyway, by the way. बताओ artificial seed, within artificial seed what will be the miniature, what will be the component? C, yes. ठीक है, zygotic embryo तो नहीं है, वो तो natural seed है. Unfertilized ovule, वो भी सही नहीं है, ठीक है. Somatic embryo. Okay, somatic embryo is nothing but The embryo is generated from the any somatic cell, from the leaf cells, from tissue, from stem cells. Okay, what you will do, you will develop an embryo, then you will enclose with a gel. Which gel? जेल में बारे में बताओ कोई, कौन सा जेल, कौन सा जेल यूज करते हैं बताओ, कमेंट में लगा दो फटाफट। Alginate, sodium alginate, exactly, good. Statement: import permit is an official document needed to introduce in India in any plant genetic resources from the other countries. The import permit is signed by the Director General ICAR. बताओ आंसर बताओ, इसमें कौन सा स्टेटमेंट सही है, कौन सा गलत है? हाँ, सोडियम आल्जिनेट तो सही है। आ जाओ नेक्स्ट क्वेश्चन में आ जाओ। ओके, you can see here, import permit is official document. It is needed for the import of any material from the other countries. Import permit will be issued or signed by the Director General ICAR. अरे भाई, हाँ, आ गए आप लोग। -5, कौन कौन ऑप्शन ए बता रहा है? सब लोग -5। कहाँ आएगा इसे आंसर? हाँ, सी तो आंसर सही है बच्चों, सी उधर ही छोड़ दो। मैं कौन कौन ये लगा रहा है ना, उनको बता रहा है, ठीक है? मैं आई हैव ऑलरेडी टोल्ड दैट, ओके, द प्लांट ब्रीडिंग सेक्शन विल बी वेरी इजी, व्हाई यू आर मेकिंग इट कॉम्प्लिकेटेड? You should know these basic things, okay? For import of any material, you should need 2 certificate. One is import permit, ये तो जनरल क्वेश्चन आएगा, ठीक है, आता रहता है, इट इज वेरी इंपॉर्टेंट, दैट्स व्हाई. And you should need phytosanitary certificate. Import permit from Director NBPGR. Okay, he will give the import permit. Your phytosanitary certificate will be issued from the export country. Okay, this is the important thing you should know. Sorry. Yes, Madhu. Phytosanitary, yes. Director of NBPGR. There will be a quarantine once you import the material, okay, through the NBPGR. It will go for DPPQS. There will be a quarantine there for 40 days or 50 days based on the material. Then you will get anything. Okay, who will in India, who will monitor export of any germplasm? बताओ, लगा दो आंसर लगा दो। हाँ, फरीदाबाद, DPPQS फरीदाबाद। yes, प्लांट क्वारंटाइन ऑर्डर 2003 शायद, एक बार देखते, मतलब उसी एक्ट में सारे क्वारंटाइन होता है, वही है ना, प्लांट क्वारंटाइन ऑर्डर। यस, प्लांट क्वारंटाइन ऑर्डर 2003, यू शुड नो दैट आल्सो। अभी बताओ, हाँ, हो गया, इधर आ जाओ। एक्सपोर्ट के लिए मतलब कौन सा थ्रू एक्सपोर्ट होता है? इंडिया थ्रू. If anyone from other countries they want to access the any germplasms, any genetic resources from India, NBPGR, अरे भाई, रिमी, yes, good, नेशनल बायोडाइवर्सिटी अथॉरिटी। कहाँ है नेशनल बायोडाइवर्सिटी अथॉरिटी? is located in Chennai. These are the things you should know, okay? If any one out of the country, any foreign person, if they want to access the germplasms or genetic resources in India, okay, they should get an prior approval from the National Biodiversity Authority, which is located in Chennai. Okay, these are very important for you guys. आ जाओ आ जाओ। अरे ये तो बहुत इजी है, बताओ। इसे वो तो मुझे लग रहा है 12th का कुछ एग्जाम है, बेसिक एग्जाम। 12th में भी कुछ डिफिकल्ट पूछता है। राइस, गोल्डन राइस, टोबैको फर्स्ट जीएमओ, टोमेटो सेल्फ लाइफ, कॉटन जीएमओ हाइब्रिड, ये डायरेक्ट शूट। देखो ऐसे आप देखना है। राइस के लिए देर विल बी, जस्ट यू कैन इजीली टेल दैट देर विल बी गोल्डन राइस। ये 3, देर इज नो ऑप्शन फॉर 3, go for 3, just complete it, skip it, but don't skip here. Okay, I am giving some information. Okay, ये तो क्लास में दिया था, मतलब one who are attending my classes in the live classes, they already know this fact. Okay, the one who don't know, which are one who are attending the YouTube live classes, you can just see. This is the normal rice, this is golden rice 1, this is golden rice 2. What exactly the golden rice? In naturally in rice, okay, there will be a no pathway for the beta carotene production. Your beta carotene is nothing but provitamin A, precursor for provitamin A. Why you need vitamin A? Okay, without vitamin A, you can see the blindness. Okay, there will be so many deficiency symptoms without provitamin. Sorry, without vitamin A. Okay, the color, blindness. Okay, so to overcome that, okay, Ingo Proctricus and Peter Bayer, okay, they have started this particular idea, which is golden rice. This is the normal pathway, GGPP, geranyl diphosphate, okay, the basic pathway in rice. Okay, if this GGPP will turn to 14, you need a one gene, that is PSY. For you guys, the one who are preparing for ICAR PG, there is no need to see all the genes and enzymes. Okay, there is no need to go through all of this, but you should know the basic idea. Okay, there will be a three enzymes basically. Okay, it will naturally, they will be not in the rice. Okay, the PSY phyton synthase, it will convert phytin to lycopene, that is your CRT1, lycopene to beta carotene. You should know the what exactly beta carotene. Beta carotene is nothing but provitamin A. In rice, naturally it will not present. So to convert GGPP into beta carotene in case of rice grains, you should need 3 enzymes. Okay, these 3 enzymes will produce from these genes which are present in the daffodil and bacteria, that is Pentoia ananetis. Earlier it will called as a Erwinia iridivora. Okay, your PSY gene from the daffodil, your CRT1 from Erwinia. Okay, the 2 important genes. In case of golden rice 2, okay, in case of golden rice 2, the PSY gene, this particular gene, we took from maize, not from daffodil. Okay, we took from maize. That is the difference between golden rice 1 and golden rice 2. The PSY gene in case of golden rice 1 is from daffodil. The PSY gene in golden rice 2 will be from maize. You should remember that particular thing. How it started in 1996, 1990s, it will start from Ingo Patricus and Peter Bayer. Then golden rice 1 developed. Okay, that is from daffodil PSY and bacterial CRT. Then golden rice 2, that is your maize PSY gene. Okay, then in Philippines is the first country to commercialize the golden rice. This is the thing you should know. Okay, clear? Golden rice. The basic idea, there will be no natural pathway for the beta carotene, that is for provitamin in case of rice. So we took some genes from daffodil and maize and bacteria, so that in that particular pathway it will convert into provitamin A. That's the basic idea. PSY1 for golden rice, what is daffodil? PSY1 for golden rice is from daffodil, exactly. For golden rice 2, we took the same gene from the maize. Naturally in maize there will be a pathway. Okay. Your tomato, flavor saver tomato. Okay. Natural tomato, what will happen? Polygalactonase enzyme. Okay, you should remember this enzyme, polygalactonase enzyme. What it will do? It will soften the fruit wall, okay, and the tissue around your fruit, so that there will be a short shelf life. Okay, your fruit will get softened and it will damage. What we will do in case of GM, we will use the antisense RNA technology. We will inhibit that particular polyglucoronase enzyme with the antisense RNA. What it will happen? That enzyme will be inhibited so that the translation of that particular mRNA will inhibit the production of polyglucoronase enzyme. So less polyglucoronase enzyme produced, you can delay the fruit softening. There will be delay in fruit softening, so there will be a long shelf life. You can see polyglucoronase enzyme will be production reduced and delayed softening, longer shelf life. First genetically modified food crop commercially used, that is your flavor saver in 1994. Antisense RNA, is it genetically modified? Yes. अरे भाई ये सारे genetically modified है आपका GMO हाइब्रिड, GMO फॉर सेल्फ लाइफ, GMO फॉर विटामिन, दोस्त राइस टोबैको कॉटन में आते हैं ना सारे, शुरू में सारे GMO से, इसलिए मैं GMO के बारे में बता रहा है। और एक और आपका कॉटन तो पता है इंडिया में तो बोलगार्ड 1, बोलगार्ड 2 ये सारे है ना हाइब्रिड से, ठीक है। हाइब्रिड में हम लगा दिया वो क्राइजीन, ओके। इन बीटी कॉटन, यस वही बता रहा हूँ, इन बीटी कॉटन, ओके, what we are using, those are hybrids. That's why GMO for hybrid. And tobacco, the first GMO which is developed, okay, for antibiotic resistance. The first crop which is tested for GMO for transgenic is your tobacco. You should remember, okay. Monsanto and Myco collaborated. अरे भाई सही से बता रहा हूँ। अभी there is no Monsanto, okay, it is merged with Bayer crop science, okay, there is no Monsanto now. Flavor saver हो गया, match the following, ले लो 4 marks easily. yes Kavita, it is now Bayer as I told. B C कौन लगा रहा है भाई? अरे इक्रिसैट तो आपको पता है 2, C क्यों जा रहा है, इधर ही आ रहा है ना। सीमिट तो पता है, वीट D1 ले लो, सीमिट वीट D1, आपको इक्रिसैट भी पता है, पिजन P A2 ले लो, आपका ऑप्शन बी, ठीक है। ICAR International Crops Research Institute for the Semi-Arid Tropics, mandate crops, sorghum, pearl millet, chickpea, pigeon pea, groundnut. You should know this basic idea. अभी बताओ एक और क्रॉप रिसेंटली उन्होंने उनका मैंडेट क्रॉप्स में डाला है। रिसेंटली दे हैव इंक्लूडेड वन ऑफ द क्रॉप टू देयर मैंडेट क्रॉप ICAR में, इन ICAR. Sorghum, pearl millet, chickpea, pigeon pea, groundnut. Which is the another one? Anyone from Telangana, Andhra, Karnataka, they should know this. RPH 72, वो कौन सा क्रॉप है भाई? बताओ जल्दी। Yes, good. वर्षा कुलकर्णी, finger millet. Captain Castle, ragi. Yes, they have included finger millet recently. These are the things you should know. IIT, where it is? In Patancheru, Hyderabad, okay. IIT, International Institute for Tropical Agriculture. Major mandate crops are cassava, yam, maize, soybean, banana, and cowpea, which is located in Nigeria. CIAT, International Center for Tropical Agriculture. Beans and cassava are again important here, okay, which is located in Colombia. Next, CIMIT, International Maize and Wheat Improvement Center. Wheat and maize, where it is? In Mexico, okay. There will be so many achievements from CIMIT, okay. your Kalyan Sona, Sonalika, which are from that particular institute. Okay, the green revolution for wheat. It's a shuttle breeding. The famous shuttle breeding. They have used two location. Okay, that is from CIMIT. There will be so many biofortified CIMIT lines in wheat. Again, those are from CIMIT. Okay, this is the major institute. IRRI, Indian Research International Rice Research Institute. Where it is? International Rice Research Institute. Your IR-64, IR-8, IR-36. Okay, which are from Philippines, Manila, Philippines. Yes, good. The acronym for ITPG RFA. ITPG RFA. Again, it is very important. They will target these kinds of question. ITPG RFA, PPV FRA, CBD, okay, NBA. These are the things you should know. Okay. फिलीपाइंस मनीला अरे इसको हाँ बी भी बता रहा है क्या हाँ इंटरनेशनल ट्रीटी फॉर प्लांट जेनेटिक रिसोर्सेस फॉर फूड एंड agriculture. Okay, this is your option. We will see something about ITPG RFA. It is adopted in 2001, entered into force into 2004. You should know again. You should keep in your mind. Okay, it came into force in 2004. Okay, under the United Nations. Okay, the objectives are conservation and sustainable use and fair and equitable sharing of that benefits arising from their use. Okay, the main thing is conservation and sustainable use of plant genetic resources for food and agriculture. Okay, and the fair and equitable sharing, which has the benefits which are arising from this particular genetic resources. Okay, we should share that benefit between the countries. This is what exactly your ITPG RFA will tells. Benefit sharing fund, money collected from the commercial use of seeds is invested in the projects worldwide with the small scale farmers. Okay, we will help the small scale farmers directly or indirectly with some infrastructure or any facilities. Okay, from that benefit sharing fund. Again, this is the important crop. They have mentioned in annex 1, they have mentioned 64 crops. Okay, out of 64 crops, how many food crops and how many forage crops? Batao koi? How many food crops and how many forage crops? Around among 64. In ITPG RFA, they have mentioned 64 crops. Out of 64 crops, 35 food crops, 29 forage crops. Good. कौन बताया था? Ananna Patra. Very good. Food crops, they have mentioned there will be different food crops, right? Wheat, maize, potato, cassava, beans, banana, like that. Forage crops, legumes, grasses. There will be so many alfalfa, clover. But they have mixed soya bean, tomato, and sugarcane. Okay, they have not mentioned these three crops. Okay, under the ITPG RFA. Okay, in the 64 crops. ठीक है, लास्ट क्वेश्चन. इसके बाद एक स्ट्रेटजी मतलब एक टिप आप ऐसे मान सकते हैं. Chronological steps in the development of genotype and the release of a crop variety. In the last previous year question paper also there is one question regarding this. I've already explained it, complete procedure. Okay, how we will develop a variety, how it will enter to the seed chain, how means release chain. Okay, I've already discussed about it. You can go and watch the 2020 PYQ discussion of mine. अभी बताओ, आंसर बताओ. First what we will do? बी डी में ऑप्शन नहीं देखते, पहले इधर आ जाओ. First step is your genotype development. B. After genotype development, you should go for preliminary yield trial. That is in your station level as a breeder. B. A. Okay. After this, you will go for multi-location trial. C. Then after that, you will go for identification. then you will go for notification. BACED. BACED. This is your right option. B. Okay. D कौन बताया था भाई? D. First you are going directly. You are going for identification. अरे पहले तो डेवलपमेंट तो करना है ना जीनोटाइप. After that you will identify ना. ठीक है? Simple procedure. Okay, I will tell. First you will develop. How we will develop from different sources. Okay, from ICAR system, from state agricultural universities. Okay, SAUs, from private breeders, farmers. Then you will go for station trial within your station. Then you will go for multi-location trials. That is your first one. First year you will go for IVT, initial varietal trial. IVT. Then you will go for AVT 1. AVT 1. Then you will go for AVT 2. Okay, there will be 3 years of multi-location trial. What you will do next? After that, you will identify variety in AICRP meetings or workshops. Okay. Who will identify? There will be varietal identification committee. They will identify. Then after this identification, you will, as a breeder, you will submit a proposal, okay, for the release. After identification, there will be release. Okay, who will release? Your DDG crop science. So DDG crop science will release the variety. Okay, he is the head of Central Subcommittee on Crop Standards Release and Notification. Central Subcommittee on Crop Standards Release and Notification. He is the headed by DDG Crop Science. Okay. Then after release, again you will submit for notification. Who will do the notification? Central Seed Committee. Okay, which is under the direct under the Ministry of Agriculture and Farmers Welfare. Okay, they will notify the variety. Once it is notified, it will comes under Seed Act 1966. It will comes into the seed chain. Okay, the foundation seed, certification seed. Okay, the rules and regulations. It will comes under that. After notification, the final will be the gazetted notification. Okay, notification is nothing but the gazetted under the Indian government. Was there any class yesterday, sir? अरे भाई मैं second video है बस, ठीक है. 2020 का खत्म किया है. It is 2021, the second class of mine. Okay, from tomorrow there will be continuous classes. This is all about today's paper. Simple paper. There are 20 questions. There will be simple again. You can easily answer all of the question. Okay, if you have some basic idea, 100 marks easily. Okay, tips. Assertion and reasoning में मैं already बताया था. Okay, in assertion and reasoning kinds of question, if you are getting a confusion, okay, and if you think that there will be some mistake in the question while setting the paper, maybe he confused whether he is setting and both are not correlated, but you are thinking that there will be something which is connecting, or there is a connection between those two, and you are also thinking that both are right, means correct answers, correct statements. Okay, just means anyway you are already decided to choose one of the answer. Okay, at that kind of time, okay, go for first option. Okay, that means he is trying to give an assertion and he is also trying to give a reason to support that assertion, but while framing the question or in that procedure, maybe there will be some confusion or mistakes in the question. At that point of time, you just go with the first option. Okay, but when there is a problem with the question and you are identifying there is some incorrect, okay, the statement is not correct, it can be easily identified. At that kind of scenarios, you can go for other options. You can see that R is false or A is false. You can easily identify the false statements. But in some of the times now there will be a confusion that yeah, it is looking like the both statements are right, but there will be some gap or there will be some grammatical mistake, or while framing that questions, okay, there will be some mistakes. That kind of scenario, go for A option. Sir, please mention a schedule for plant bidding in group. किस ग्रुप में बताओ भाई, कौन सा ग्रुप में बताना है? I have already telling in this particular class there will be a continuous classes from today up to 2025 paper. What kind of schedule you want? That will be in the exactly at 10 PM. This is the schedule 2/2. ठीक है? 10 PM शेड्यूल वही है ना, मैं बता रहा हूँ, आज से 2025 पेपर तक कंटिन्यूसली क्लास लगेगा, ठीक है, 10 PM एवरी डे ब्रीडिंग का. I didn't understand, sir. Karthik, what you want to know? Can you repeat? Which question? What you didn't understand? Tell some tips daily, sir. अरे भाई, टिप्स तो मिलता रहेगा, ठीक है. अरे मैराथन मैं क्या करूं? मैराथन भी चाहिए, एक के बाद एक जाएंगे ना. मैं बताया था मैराथन भी इसके बाद मैराथन ही होगा, ठीक है. थोड़ा सबर करो. अरे भाई फिर से क्वेश्चन, आर यू रनिंग ए मैराथन सर? नहीं भाई, मैं तो नहीं भाग रहा है, ठीक है. मेरा उम्र तो हो गया, आप लोग भागो भागो मैराथन. ठीक है, this will be the end of this session. We have discussed 2021 paper. So tomorrow I will come up with 2022 paper. Again, that is very important. Okay, by seeing these papers, you will also get some confidence and some basic information or basic idea how to attend the questions. Who will be dealing marathon, sir? You or Mosam ma'am? कौन चाहिए आपको? साजन कौन चाहिए, मौसम मैम और मैं मैराथन के लिए? आप बताओ. यू ठीक है. मैराथन में मैं ही रहेगा, ठीक है. थोड़ा एडजस्ट कर लो, हिंदी और इंग्लिश दोनों में चल के जाना है, ठीक है. मतलब वेयर यू आर नॉट गेटिंग, यू जस्ट लेट मी नो, वी विल ट्राई टू क्लैरिफाई द थिंग्स. ओके, मेरा हिंदी थोड़ा ऐसी है. कॉन्सेप्ट तो समझ आएगा 100%, डोंट वरी. टिप्स तो कुछ नहीं है भाई. टिप्स कौन कार्तिक पूछ रहे हैं ना, मैं वही बताया था ना assertion रीजनिंग में. If there is a confusion, if you are not getting exactly what they are telling, okay, and you are also not able to identify the incorrectness in the particular question, that means your seventh sense it's telling that there is something right. At that point of time, you should go for option A. Okay, this is the thing. Always don't go like that. Okay, when you are already decided to attend that particular question, but you are stuck with that question. Okay, you are not able to find the incorrectness. You are not able to choose this thing false. A is false or R is false. You are, that means there will be, you are thinking that both are right statements. At that kind of time, and where R is not the correct explanation of A, you are not able to choose this also. Sometimes, okay, you are, there is a something, it is explaining. Okay, there will be sense na, seven sense, it will tell us like that. At that kind of time, you will just go for A option. Your Hindi is very good. You only want it. Hindi understood because attending live class of aggregate pathology continue with the breeding. मौसम मैम, कार्तिक के शर्मा को मौसम मैम चाहिए। ठीक है, थैंक यू सर। ओके, this is all about today's class. We'll come up with the next paper in the upcoming class. Thank you for one who all who joined this session.